Jessica, actually, you're not supposed to be talking for 50 minutes. It's only...
it's 30.
30 minutes, yes.
I'm supposed to be talking for 30 minutes.
30 minutes, not 40 minutes.
I know. I don't really want to talk about 40 minutes. Fine.
Up to you, Jessica.
Okay, thanks. Okay, so I'm going to touch the other open question that Anil discussed. So the first one was, how do you test significance? How do you deal with train and test? And the second one here is, how can you try to interpret the models that you and, because they are different from univariate models, so what cognitive neuroscience conclusions can you make about them? So we've seen this, this is the multivariate analysis framework. You have data, feature selection, model specification, accuracy and significance. So Anil and Jonas went over most of it, and I'm now going to go into the model interpretation. So most of you have this question, which is where in the brain is the information of interest, which means which brain areas contribute most to the model's prediction. Anil has shown you that we can compute weight maps. So from linear methods, either kernel or not, they end up in attributing one contribution per feature that you enter. If you have image, as in most cases, you might end up with one weight per voxel and build what we call a weight map. So you see here I put the solution, the mathematical solution, but basically what you end up with is one weight W per voxel. And this is an example of a weight map from the HAXB dataset, faces versus buildings, with using an SVM classifier, just using the whole brain pattern. But what actually is in those weight maps? Can we threshold them? Can we just look at the ones with the largest contribution like we would do with an SPM? Well, unfortunately, the answer is no, we can't. Because all of the weights are used in a linear combination with the signal to make the final prediction. So you see the formulation there. If you have a new data point xy, you're going to combine the signal in xy with the model parameters to make a prediction f of this new data point. So we can see this like intuitively as an election process. You have like different little people here. You have one that some of them have more weight than others. They just, I don't know, they cheated and then they can vote multiple times. And let's say that one of them has 10 votes for the blue class, one of them has 8 votes for the red class, and then you have like people with only 1 vote and they vote for one or the other. If we look only at the people with the highest, like the number of votes, then we're going to say, okay, we have one predicting for blue, and then he has 10 votes, and one predicting for red, he has 8 votes. So you are basically looking at the decision function being, like the prediction being blue, because the blue guy has more votes. However, if you look at all the people, and you count them all, you end up with a prediction for red. So if you look only at the highest contribution, this does not represent the model and the prediction that you're making from your data. Another issue with the weight maps is, what do those parameters actually represent? Is it only, like, we know it's discriminating between classes, or fitting the data, it's what we ask from it, but how are those weights computed? So if you take a very simple example with only 2 voxels, and let's say that in voxel 1 I have my signal of interest with I don't know, higher activity for class 1 and lower activity for class 2. And then I have some noise signal, dn, which might be the heartbeat. And in voxel 2, I have only that noise signal. So what is my classifier going to do if I want to discriminate between class 1 and class 2? Well, if he wants to get a very nice discrimination, he's going to take voxel 1 and take out the noise by subtracting voxel 2. And so we end up with a weight of 1 for voxel 1 and a weight of -1 for voxel 2. And usually we just look at the magnitude of those weights, and so we end up with the two voxels having the exact same weight into the decision function, although our information of interest is only into voxel 1. So we really have to be aware that the weight maps that we are seeing do represent the discrimination between the two classes, knowing the signal and the noise. So there has been work to find a solution for that by Halford et al. In 2014, where they basically reconstruct weight patterns from what they call the weight filters, which are those weight maps that I just shown you. I'm not going to go into the details of the paper because there's still kind of a discussion on how far their conclusions actually should extend, but it should definitely be looked into and maybe try to look at both the weight patterns and the weight filters to see where your information could be located. So here I'm going to discuss a few of different strategies that have been established now to try and look at where in those maps is the information of interest. It's not an extensive list, and I'm just showing the main IDs and not getting into each method in specifically, but so that you get a few leads. So I'm going to present 7 of those IDs, and they actually come at different steps of this framework that I've shown you about multivariate analysis of the data. And so you have 3 steps at the feature extraction and selection. And then you have 2 other strategies that are directly implemented within the model specification. And then you have finally a posteriori interpretation of the weight maps after you have computed your model. So first, and you probably know this one, it's just the selection of a region of interest. So you have a hypothesis, and you say, okay, I want to look at the discrimination between faces and buildings, and I want to look at the fusiform face area because I think the information of interest is there. So what you can do is you build a model choosing only that region of interest, and then you can look up at the accuracy, and you can compare that accuracy to a whole brain model. And basically, if the accuracy is higher or equal to the whole brain model, then you can kind of, not super confidently, but still say that most of the information of interest is located in those regions, or at least pretty well represented in those regions. So you are not losing discrimination by selecting only these regions compared to the whole brain. On the other hand, if you have a lower accuracy using only those regions compared to the whole brain, then maybe the information of interest is more distributed than you thought. So here we're just looking at the accuracy, so we're not looking at the weight pattern or the weight filter. And it's anatomically or functionally relevant. However, well, you do need to have a hypothesis, so either you need to read beforehand, but it's not very data-driven. So you still have, as we see here, we still have a weight filter for that we can see for each voxel, and we still cannot interpret that. And then you have, of course, linked with your hypothesis, an arbitrary choice of the region number, F size, and usually you might want to test multiple regions of interest, and then you end up with a multiple comparison problem because you are testing for the same effect in different regions of interest. So still at the feature extraction level, we have the searchlight technique, which is also pretty well known now and used by Krieger-Skort et al. In 2006. So what is this technique doing is it's going to select one voxel and its neighboring voxels. You define like the radius of the neighborhood is user-defined. And then it's going to compute a model for the discrimination you are looking at, and you obtain an accuracy, and it's going to report the accuracy in a new map for that specific voxel. And then it's going to do it for all of the voxels. So instead of having a weight map, you obtain an accuracy map that you can then threshold based on the significance, for example, of the accuracy. So it's not a weight filter, which is a good thing, and you can obtain a threshold map. However, these are spheres, so it is not anatomically or functionally relevant. It is also locally multivariate because you are just using a few voxels, neighboring voxels, to make your prediction. So if some of you are interested in networks, for example, then you will probably not be able to identify any of those. It's the user choice to define the neighborhood size, and it influences the results. You end up with, of course, one model per neighborhood, which means a multiple comparison problem that you need to correct for. And yeah, it can be pretty time-consuming because you need to build one model for each neighborhood. So there's a nice review of the pros and cons and potentials of the Searchlight by Edsel et al. In 2013. So the last, now we're not into feature extraction anymore, but into feature selection. And so in here, we're going to first rank the features according to a specific criterion and select a subset of those features to build our model. So this can be done in the various ways. You can have a very simple example like the t-test. You just rank your features and according to a test with your labels, or you can do a correlation test, whatever. Then you have the recursive feature elimination, so you can build an SVM, compute the weights, and then eliminate the voxels with the smallest weights and do it over again. And then you select, in the end, the subset that leads to the maximum accuracy. So this is definitely not an exhaustive list. But what is good with feature selection is that it is data-driven. But then you obtain a map that is sparse in the voxel space. So it can be kind of a salt and pepper. So it's not necessarily very anatomically or functionally relevant. It is time-consuming as you need a nested cross-validation. It depends on the selected technique. So if you have a flow in your feature selection technique, then it's going to be reflected in your model power. So if you are using a multivariate way to select the features, then you might end up with a set that is not very representing the maximum power you could have from a multivariate analysis. And then different strategies might lead to different patterns, although with the same accuracy. So related to feature selection, doesn't work. Excuse me, it doesn't work. Yeah, this one, thank you. Okay. So related to feature selection are sparse algorithms. So basically, the sparse algorithms are going to select a subset of the features directly when estimating the model. So this is based on regularization constraints, and you can have different regularizations like the lasso or the elastic net that are going to force certain weights to be perfectly null. So what is good with sparse algorithm is that as for feature selection, it is data-driven, but this time it's embedded in the model. You end up with the same maps, basically, which are sparse at the voxel level. And then you also have the fact that different regularization terms might lead to different patterns for the same accuracy. So there has been work on that, especially for stability selection by Baracco et al. And by Rondina et al. So either using clustering approaches or by using simply resampling the data set and selecting the voxels that have been selected consistently across like slightly different versions of your data set. So also a sparse technique, but I wanted to just show it because it can be a hierarchical approach. So Anil has already discussed it. I'm not going to spend too much time on it, but just so you can see exactly what it is. So it's the method from Filippone et al. In 2012. It's based on a multiple kernel learning algorithm, which is sparse. So here, instead of building only one kernel for your whole brain analysis, you take your image, you apply an atlas, and for each of the regions as defined by the atlas, you're going to compute a kernel. And then these kernels are going to be combined, and their contribution to the final decision function is going to be sparse. So you have an L1 constraint on those kernel contributions, dm. And so here is the mathematical formulation. We are using the Rakatoma-Monji et al. 2008 simple MKL algorithm. And what we can, I'm just going to show you the image here. Yeah, so we can build a map of the contributions per region. So we can build two things, actually. We can build the map of kernel contributions DMs that you can see here also for the Hagsby data. And you see at the bottom right, you have a histogram of the M, basically. And although we input 116 regions, only 12 or 13 have been selected in the different folds in average, and the others have a perfectly null contribution. So you can decide to just look at that list of regions, and that would represent what your model is about. But as we do have those contributions, we can still build the WM, so we can still have a look at the weights in each of those regions. We can look at whether they are relatively homogeneous or not. This, all in all, can be seen as a relatively hierarchical model because you have weights at the region and then at the voxel level. So what is good about this technique is that you're using an atlas, so it is anatomically or functionally relevant depending on the atlas you're choosing. You use the full pattern to build the model, so you're not losing any multivariate power. You can obtain thresholded in the sense that there are perfectly null contributions in the list of regions that you obtain in the end, and it gives a hierarchical view of the model parameters. On the other hand, it is a sparse technique, so the stability of which regions are selected should be investigated. For example, here at the very bottom of the list, you have region 10, which is occipital right, which has a very low weight, 0.6%. And it's probably because it was selected in some of the folds of the cross-validation, but not in all of them. Maybe it was selected in only one fold. And this depends on the constraint of the MKL. Here it's an L1 constraint, so it's not going to select regions that have correlated signals. And so you definitely want to maybe look at the stability of which regions were selected. And of course, it's going to depend on the atlas you choose. So now we are at the model step. So we have built a whole brain model and decided not to do any feature selection, not to do any feature extraction, and we just want to look at the, let's say, the weight filter and see whether we can do anything with it. So the first thing we can do is a permutation test. So for each of the permutations that Anil said you should build to maybe assess the model significance in terms of accuracy, well, you can also build a weight map. And you can build 1,000 weight maps, and then for each voxel, look at whether the weight in your true model is larger than the weight in your 1,000 permutations. And it basically comes back to doing an SPM univariate analysis on the weights. You can obtain a map that is thresholded, and you have used the full pattern to build a model. However, this is computationally very expensive because in the end you still need to build as many permutations as, of course, you need for assessing the model performance, but you still need to take into account that, that you end up with a multiple comparison problem because you are looking at the significance at each voxel. And the correction for multiple comparison seems to be different from the correction usually applied in SPM. Maybe, so in their paper, Kankar and Davatsikas say that maybe the FDR or the PR4NI are way too conservative for those weight maps that we obtain. There are less false positives in those weight maps. So it is not clear which correction should be used. I said it's very expensive in terms of computation, and in their paper they have come with an analytic approximation of the null distribution for an SVM classifier that only applies for SVM though. And then the last thing, which is a very simple thing to do, is just take the atlas after you have built your model and average the weights within each of the region specified by the atlas. So here is just taking the absolute values and averaging. So you can also obtain a list of the regions with the contribution, but you can see that the plot that was sparse earlier with MKL is not sparse, and so you cannot threshold that list. And of course, it still depends on the atlas. So I've been talking about exclusively neuroimaging data, but all of this also applies for other types of data. And you can see here an example of the MKL that was built using the different channels. So you have one kernel per channel, and then you can look at the weights of each of those channels at the contribution of the kernels, basically. And here it's plotted, and you can see that you have some perfectly null contributions, like represented by the white dots here. And I've also been talking exclusively about linear models, but how about nonlinear models? So there has been a way devised to get maps for nonlinear models, so statistical sensitivity maps for some classifiers, not all of them. And basically the sensitivity map of a linear classifier ends up being the weight filter squared. So they're very related. But then should we be using nonlinear kernels in neuroimaging? That's another question. There's a paper by Kriegel et al. In 2012 that said that maybe for some local cases it might bring information, and that maybe you can look at the whole pattern in a sense of linear way, and then if you want to investigate more specific regions, maybe non-linear kernels could bring more information, but it's still an open question. Okay. So in conclusion, interpreting machine learning-based models can be complex. Different strategies exist, but it's always a trade-off between different parameters, whether or not you want a thresholded list of regions or of voxels, if you want anatomical functional relevance, how you want to use the multivariate power, the stability of the pattern, and of course, it's a practical detail, but still the computational time. So it is now, hopefully, your informed choice, and on that, I leave you with a list of reading. Thank you.
Questions? Yes. Can you maybe stand up for the microphone? Or?
Yeah, can you come to the microphone for questions, please?
Hi. So it's pretty clear to me why the weight maps you get from MBPA straight out the box are maybe difficult to interpret. But it's not clear to me why the weights that come out of MKL don't also suffer from that problem. So is there some careful way to do thresholding of those weights, you know, for what you described for the method, or is there for some reason you don't need to worry about it in that case?
So the weights are as difficult as they are to interpret. It's more that using the MKL on regions, you have then a sparse selection of the regions. So you can, instead of looking at sparse voxels that can be like salt and pepper maps, then you can just look at the list of regions that you decided have an anatomical or functional information or relevance. So, but if you go down to the voxel level map that you can derive from there, you end up with exactly the same difficulties that you have in the beginning. Just that it's sparse in terms of region. So instead of having salt and pepper maps, you end up with some areas with weights and some areas with no weights. But it doesn't make it easier in that sense. It's more in the list of regions you can obtain from it.
Thank you.
You mentioned the use of different atlases of functional regions at several points. Could you talk a little bit more about some of the major options, along with some advice about how to choose between them?
Sorry, can you repeat?
Yeah, I was just asking about the use of different atlases that you had mentioned a few times, particularly functional atlases. Yeah,
so I'm not really in the field of choosing an atlas, basically. I'm using the AL. I'm actually not really using the MKL that much. We've implemented it, but it's more because I'm not a neuroscientist, I don't really have that input. But there are a lot of atlases. There are a lot of open that you can actually download the atlases from different websites, and they can be either based on the anatomy of the regions or based on different functional analysis like connectivity and things like that. But yeah, they usually include like around 100, 200 regions. I don't know if there are others that you want to cite.
Yeah, it really depends how you think your brain should be split into separate regions, which is sort of your contribution. You put that as your priority. You could say just use the main lobes of the brain, and I would split the brain in 8 regions. If you think things are really just split that way, or you go down to much smaller brain regions and maybe have 100 or 200 regions, which is quite arbitrary. And it's really your choice, and different atlases will actually lead to different selection of weight. And then it's difficult to compare one model with the other than maybe using like the area under the curve sensitivity. The compromise between specificity and sensitivity would be one way of saying maybe with this atlas I get better result than with the other one. It's hard to say in advance what will work and what will not work.
Yeah, well, actually, when we say an atlas, it's you inputting information and about how to divide, and you could even like decide to get the like main networks, like resting state and networks, and say, okay, I want to build one kernel using those 3 regions from the DMN, and then and see which part of the networks or which networks would be selected. I mean, it's really, we use atlas, but it's just a way of how you want to separate the brains.
Thanks.
Hi. I was wondering if in trying to define voxel subsets that have a special contribution to the prediction, to the classification, are there established methods for exploiting how pairs of voxels contribute together, so that you could take one voxel and then for each, but then separately permute every other voxel, and then that gives you a sort of distance between two voxels that says something about their joint contribution to the classification, and then essentially you have a distance matrix for the whole brain which you can then parcellate. Are there approaches like that?
Well, you kind of like come back to a pairwise comparison and more like connectivity linked measures than using the whole brain pattern. If you are looking just at the like how two voxels are correlated, then you're not going to use the fact that you can build a linear combination of the signals in all the voxels of the brain to build, because we are still building a decision function in the end, and it's still what you're going to have from a multivariate model. If you don't care about the predictions, or if you don't want to investigate that, then maybe connectivity measures would be more appropriate than just using a fully multivariate whole brain method. I don't know if that answers your question.
Well, I was thinking you use the whole brain, but then you selectively permute one voxel, and then you show how permuting that one voxel changed the contribution of all the other voxels. But perhaps I should ask this outside this interaction, but
Do you have an answer?
I'll think about it. Thank you.
I don't have an answer right now.
Yeah, we need to think a little bit about it.
But think about